Okay, this is the last talk of this symposium in Bromeliaceae and will be given by Professor Leebens Mack from the University of Georgia. He's working in the Department of Plant Biology.
Well, thanks so much for organizing the symposium, and thanks to all the speakers for a great set of talks. And really, these are exciting times in Bromeliaceae phylogenomics. Now, phylogenomics, as I showed this the other day, I'll just reiterate, is a two-sided coin. On the one hand, we're using genome scale data to reconstruct relationships. On the other hand, we're using these phylogenies to understand genome evolution. Actually, we just heard about how this is being done for mitochondrial genome evolution. Um, I'm going to kind of recap points that have been made over the last couple of days by Tom and Chelsea and others. One of the points I want to make is that for this first side of the coin for phylogenomics, plant phylogenomics generally, monocot phylogenomics, bromeliad phylogenomics, this 353 kit and this other one as well, which I'm actually less familiar with, are quite useful. And I want to make a shout out to the organizers of the PAFTAL group. And Alex and Tom in particular, stay tuned for this paper that we just heard about this morning. Has now been accepted in Nature. It's really an exciting piece of work talking about relationships and also maybe even at least as interesting, perhaps more, looking at diversification rates throughout angiosperm history. So this 353 set, I was part of a project where we used it to reconstruct relationships within the Bromeliaceae, and the 353 kit had, kind of not so great. We didn't have such great LUTs. We had pretty poor capture rate. It might be the work was done by the first author in my lab. It might have been something going on in my lab, but for whatever reason, we didn't get such a great capture rates with that 353 set. Nonetheless, we did learn a lot about the structure of the subfamily, including with this analysis and also with the plastomes that we were able to pull out of this data set. And so there is plastome data set for Bromeliaceae in the NCBI. It might all be in genes rather than circles. But this actually, that's work that could be done, is pulling this out of the, pulling the raw data out of the SRA and making the circles. But I think that this is an important point. It is informative to see that we see parts of the tree where we have very low support, suggesting that there's very rapid diversification occurring in those parts of the tree. Jill Yardini put together this base set that was focused on particularly chalangioids, but Bromeliaceae, and we're getting more resolution in the relationships across Bromeliaceae, the family, using more loci. I would say that, if you look at these pies, the blue bits are the bits where the proportion of gene trees that are matching the species phylogeny, but they're quite a high proportion of genes, particularly in parts of the tree where we have rapid diversification, where those gene trees don't match the species tree. And I think that that's also very interesting, and it does raise questions. If we're interested in using our species trees to map character at trait evolution, and we're using the species tree, we're using the species tree, if those traits are coded, are influenced by genes that have very different histories, there's something that I think as a community, as the larger phylogenetics community, we need to start, we need to wrap our head around. There are some out there. Matt Hahn has talked about this issue quite a bit, but we should be thinking about it, not not wringing our hands and saying, oh yeah, there's all this gene tree discordance, saying, hey, there's a lot of gene tree discordance. What's what's the cause of that? What are the consequences of that? How is that influencing the phenotypic variation that ultimately we're interested in looking at? This doesn't project very well, but Tom mentioned also in his talk that he refined and expanded the Bromeliad bait set and come up with his own, the Bromeliad, their own, the Bromeliad 1812 set. This is work done by Bing Lee in Tom's lab and Jacob Landis in Chelsea's lab, really driving it. And they're getting extremely good resolution in this group that they've started looking at, perhaps across the family. With more loci, we can actually accommodate, get well-resolved species trees in the face of a fair amount of gene tree species discordance. We can, so more data gives us a higher confidence in the species trees. It doesn't make that gene tree species tree discordance evaporate. So we still need to better understand that. Part of understanding that is the other side of this phylogenomics coin is taking that species phylogeny, or maybe in some instances we know a gene that is really having a big influence on a particular trait. So in that case, maybe we go to the gene phylogeny and then map, try to use that to understand gene family and gene evolution within the context of that species tree. So for one contribution that we're trying to make for this, for being able to do this across the land plant tree of life, is this effort that is part of a Joint Genome Institute's community science project, the Open Green Genomes Initiative that we've been working on for a few years now. And the objective here is to fill in, generate really high quality, reference quality genomes for parts of the land plant tree of life where there hadn't been genomes available at this point. We got started in 2018. And so on this tree, which is again not really hard to see, so I'll zero in on monocots, which you're most interested in. The tan parts are genomes that were available in literature, high quality genomes, chromosomal assembly genomes that were available in literature. In blue are the bits that where Open Green Genomes wanted to add to fill in parts of this tree, including monocots. One last thing I should say here. If you want to keep track of how the data that's coming out of this project, as the genomes are being finalized, checked and so on, we're putting them in the phytosome pre-publication. So there's a bunch of genomes in there that have not yet been published on, and so you can go check them out and read all about them at Phytosome here. This, again, it's a community grassroots project, in some ways modeled after 1KP. Lots of people from around the world involved, and so these are all the folks that signed on initially to contribute, or came in later and have been contributing. These are the guys that are doing the heavy lifting at Hudson Alpha, Joint Genomes Institute, University of Arizona. Jeremy is the plant lead at JGI for plant genomics, and these others are doing various aspects of the genome, the data generation, the assembly, and the annotation pipeline. So this is, again, zeroing in on monocots. I'm going to talk a little bit about the Brocchinia micrantha genome that we've generated. So you can see that we're really kind of filling in parts of this, adding genomes for parts of this, the monocot phylogeny where there really was nothing previously, including quite a few non-graminid and graminid non-Poaceae poales. We've actually just recently added, with help from Lynn Clark and a postdoc in her lab, added a couple of grasses. So here's the Brocchinia genome that we've generated. And for the genomes that we've been generating recently, we've been able to generate both haplotypes. In other words, the chromosomes that are coming from mom and chromosomes are coming from dad by combining PacBio hi-fi data and hi-C data. And here this dot plot lines up haplotype 1 and haplotype 2. Their gene content is largely the same, but there is some interesting small structural variations along between these two haplotypes. There's heterozygosity that we're seeing, and the mean synonymous substitution difference for the protein coding sequences is less than 0.1. Here's, on the other hand, alignment with the ananas genome. And here we do see that there's some larger structural variations. We also know most of Bromeliaceae has around 50 chromosomes per haploid genome, but this Brocchinia that we've sequenced has 24 that we've assembled in both these haplotypes. So we've got some fissions and we've got some fusions that we see as we compare these two genomes. I should say that in both instances we can look at the distribution of variation and get a Ks. I said before was less than 0.1 for comparing the two haplotypes within Brocchinia micrantha. In this case, when we're comparing the ananas and Brocchinia, we've got up to 0.4. That's this big peak there, showing how much divergence we've had between the two species in their protein coding sequences across the genome. We also see this little bump here that represents the genome duplication that occurred in the last common ancestor of all members of the poales. So a couple things here. There's a lot of genome size. There's some, I shouldn't say a lot, some genome size variation in the family. For instance, telansioids tend to have slightly larger genome sizes than most of the rest of the Bromeliaceae. But generally, the Bromeliaceae has comparatively small genomes relative to many other families of angiosperms and monocots in particular. And so I'm going to make the case that it actually is really feasible and economical to go ahead and generate shotgun sequence for everything. And if we have reference genomes for good placeholders across the phylogeny, we could then do quite a bit of exciting work on this understand genome evolution, genotype, phenotype of relationships, and so on. So here's what we've been talking quite a bit about is bait capture in solution target capture phylogenomics. You start with a plant, you extract its DNA, you hybridize with baits, you enrich therefore your target DNA, then you pull out those target assemblies, make multiple sequence alignments for each of the genes, make gene trees, and then get a species tree. Hyde Piper is a widely used tool for going from the data to the sequence. We can do with a little bit more data. So typically, you might generate 1 gigabase of sequence per sample for target capture analysis. Given that the genome size for the average genome size for Bromeliaceae is somewhere around 500 megabases per C, if we generate 5 gigabases rather than 1 per sample, we're going to get 10x coverage for most of the Bromeliaceae. Telansioids, you might have to generate a little bit more. And so if you generate that data, you can then do an in silico enrichment, again, then make for targeted loci, make your multiple sequence alignments, gene trees, and estimate your species tree. You can also, with this 5 gigs of data, rather not target particular regions, but go into your data, map it to, if you have a reference genome, map those sequences to your reference genome, identify SNPs across the genome, and then take your SNP matrix and skip this step, go straight from going from your SNP matrix to your species tree. And Chelsea talked a little bit about this in her talk yesterday morning. So what do we get from this? And again, I'm going to reiterate some of the things that Chelsea was talking about is in addition to getting your phylogenies, we can, these are just a few of the things, where there are many things we can do. Look at your protein coding sequences, all of them, not just the ones that are included in any of these big capture kits. Make assemblies for each of your samples, reconcile those with a species tree, and understand, start to assess variation in copy number for genes of interest. Look at variation in copy number for TEs of interest. See how TEs might be influencing and dynamics might be influencing variation in phenotype, and ultimately do genotype-phenotype associations, doing kind of GWAS analyses. All of this is possible with with a little bit of this for on average 5 gigabases of shotgun sequence per sample. But you do need a reference genome in order to map those reads back. So I think that one thing that we should be working on as a community in order to enable this sort of work is make sure we've got a good distribution of reference genomes across the family. All right, I'm going to end talking about some very speculative, risky behavior, but I think really kind of exciting, and maybe a technology that might allow us to, it definitely could allow us to better understand what's going on within population, within species. It may allow us to understand radiations that are active right now. Emergence of new species, like we heard about with some of the talks we heard yesterday in this room. And this is, rather than using nucleotide sequence variation, looking at variation in methylome marks. We can't do, or epigenetic analyses. So last fall, in September, there was this paper that was published in Science on evolutionary epigenetic clocks in plants. And I really encourage folks to go and take a look at it. It's very exciting. This is work from a student and colleague at the University of Georgia and some their collaborators in Germany. And what they did, one of the things that they did was they had a population of Arabidopsis plants that they were propagating from seed to seed. They kept track for individual lineages, families for 30 generations. And then they looked at their methylomes and noticed that whereas there are a lot of parts of the methylomes, lots of methylation marks that are influenced by environment or developmental stage, there were these clock-like methylation marks that were diverging, where they see divergence in the presence absence particular points in the genome as a function of time. So that's these two panels. These they identified bioinformatically as being the clock-like in their evolution over 30 to 32 generations. And then they found that most of these, or quite a few of these, were in the gene body. So this is gene body methylation is one type of methylation, and that is where they're finding quite a bit of this clock-like methylation. There are a lot of other methylation marks that they also looked at that were definitely not clock-like, that contribute to plants' ability to respond to environmental change. And they took this fate. They were inspired by what they saw in this very controlled experiment, looking over 30 generations, and went to some Arabidopsis populations that had been expanding their range over maybe 200 years, or so in the Midwest of the United States. And they went in there, they I did their bisulfite sequencing to identify methylation marks. They're seeing that there's a very rapid rate of methylation mark accumulation in those systems, much more rapid than SNP, like orders of magnitude faster than they found for accumulation of SNP variation. And so they were able to then use these data to date the timing of range expansion from populations further south up the coast of Lake Michigan, and put those at dates of just over 100 years. So, and rather little, they use various calibrations, various approaches to doing this, and rather little variation among approaches and little variance among their loci. So I'm very excited about this. I mean, I'm not sure, we know it goes back hundreds of generations. We don't know if it goes back thousands or tens of thousands of generations. It probably doesn't go back more than 1 million generations. The stability of these marks, we'd have saturation in these marks, just much like we do when we go way back in time when we're looking at nucleotide substitutions at rapidly evolving sites. This same thing, problems we'd have here with these data, but if we're really interested in looking at how within a particular species, population, what the population dynamics are, this approach can be quite valuable and tell us, well, what individual in one population produce seed to get over to another population, get from one branch on one tree in for these epiphytic species in a forest to another branch on another tree in that forest, or another stand portion of that forest. So I'm going to leave it there. Give you all something to think about whether or not this technology might be what you think this might be useful. I'm quite excited about it. I'm really interested in doing experiments with groups like many of your the Bromeliaceae groups that are probably in the process of radiating. Orchids are another group that would be quite excited to look at. The thing is that Bromeliaceae, the genomes are small. This is going to be less expensive than in a lot of for a lot of other groups where we have genome sizes that are considerably, in some instances, considerably larger. So I'll leave it there, and I'd be happy to take any questions. And thanks again to the organizers and our folks who've been organizing all of us, the speakers, through the session.
Thank you. Question?
Awesome talk. I have no idea how one prepares tissue for doing like epigenetic work. What advice would you give for those of us who might be interested in this?
I do not know if it works on silica dried material, and I'm going to be working with Bob to figure out whether that works or not. Material that's been in the freezer for years works just well. We have to extract chromatin, extract the DNA and then do a bisulfite sequence analysis. Ovidio, have you done methyl work on silica dried material?
No.
Okay. And it works? Yeah. These are pretty stable. Yeah. Yeah, so okay, so yeah, and I wonder if herbarium samples would work. I mean, that would be another thing that would be quite cool. What, you don't think so? Yeah, I know. I mean, we're at the edge here, but that's an exciting place to be, I think.
Thank you.
Ovidio, if you have anything to add on doing working with methylomes.
I actually wanted, I always love hearing your talks because you bring these provocative ideas and so on. Thank you very much for this. So I have to think about stuff tonight, definitely again. No, but like Arabidopsis, okay, there is a clock right there, but
yeah,
do you think this is really cross? So can we bring this to non-models? Can we bring this to bromeliads, for example?
Well, in that same paper, and this is another application that could be very interesting. And earlier this morning we talked about clonal growth, and you're very interested in clonal growth. So in that same paper, there's work on Zostra, and they're looking at the change in methylation marks through clonal growth. And that seems to be working, again, in a very controlled situation where they knew how they were propagating these plants over years. They were able to reconstruct that pattern by looking at changes in the methylation marks.
Amazing.
So Jim, you proposed doing lots of 5 gigabase.
That would be for bromeliads with genome size around 500 megabases per haploid genome.
So that's sort of what's practical now and cost-wise, maybe. But I wondered what the price differential is on doing like actual high-quality genomes versus that. And how much longer you'd have to wait to actually just do that.
Mm-hmm. So, we were just doing some back of the envelope, Leonardo and I last night, back of the envelope, pricing for this sort of thing, and versus whole genome like high-C sequencing. For bromeliads, I think that we could do the whole thing for about $10,000, including some transcriptome sequencing to help with the annotation. Again, with the small genome size makes things pretty straightforward, and with this very high quality data. So we're talking about reads that are 15 to 30 KB, high fidelity. The assemblers just works really well and fast. So that, like, students could be doing this. If we could generate the data, we could say, okay, here's the data set. Let's, as a class, do the assembly together and evaluate. I think that this is possible. So that's about $10,000 again, I would say, per genome for a 500 megabase per 1C genome. That's what I'm saying. $10,000. Yeah, and the difference. Oh, I'm sorry. Go on. So to get a chromosomal assembly for a 500 meg genome is now about $10,000. Oh, so that would be for the resequencing. So right now, what it would be for just the sequencing, it's about $10, $15 per sample. Just once you have the libraries, just for the sequencing, it would be for one gigabase, or less than that even, actually, about less than $5. And so it would maybe be another. I figured this out the other day. Another like $5 or something like that per sample, relative to what you're already spending for bait capture, because you have to make the libraries for bait capture. You've got to go to all the work to do the normalization and so on, put pooling, buy the baits, do the hybridization, then do more QC. So you're not going to have to do all that stuff. You're just going to make the library. You will QC it, then shotgun sequence, and then spend time. And there'll be perhaps more personnel costs, because there's going to be more time and work involved in doing the in silico capture. For data generation, it is a very small additional amount, actually. Yeah. Tom?
Just to clarify, the 10,000 is for the reference genomes.
Yes.
Will be spending much less,
much less. Yeah,
of order 100 or so dollars a throw
exactly.
to get other samples. And we've talked about doing whole genera,
Exactly.
forgetting about all this target capture crap.
Exactly. That's what I'm trying. And Chelsea made the case for this. You made a case for this in both of your talks. I think that it's really not that more expensive than doing bait capture, and we've get all these other types of analyses that are enabled, particularly if we have a reference genome.
Any other question? Thank you so much.
Thanks, everybody. Let's go out and have some beers and talk more.